about half of your body's mass comes from water. And we're constantly changing the volume of water in the body as we drink water. We get rid of water as we sweat it, as we urinate. Water is constantly in flux in the body. And the concentration of substances in the water is really important for lots of physiological processes that we've talked about. So as you might imagine, the body has control systems that try to maintain a constant volume of water in the body. So that's what this lecture is about. So we're going to be talking largely about the urinary system, but also the volume of water greatly affects the circulatory system. We've touched on that before. And this being a control problem means, not always, but in this case it means that the central nervous system, a region of the brain in particular, is going to play a role. Now, when we think about the regulation of substances in the body, it's often useful to think about its simple arithmetic, and that's articulated by what's called the law of mass balance. So the law of mass balance can apply to really anything in the body. It could be energy, dissolved oxygen, etc. And it's just quite simply the amount of it that you have in the body as being equal to what you currently possess, plus whatever you take on, you produce or consume. In the case of water, this would be a glass of water would be included there, minus what you get rid of. You excrete, like when you urinate for water, or metabolize, which doesn't really apply so much for water. So the question for this lecture is, how is water volume regulated? What is the control system that maintains that volume at a relatively constant value? So like we've done for other control systems, we're going to start on the efferent side of things because we're familiar with that end of the control. And we're going to talk about the kidneys, and in particular the nephron. And as we've already discussed, the collecting duct of the nephron is the final tubule that performs the function of tuning the osmolarity of urine, or the concentration of urine, according to what the body needs. Does the body need to hold on to water and therefore produce a more concentrated urine? Or is the blood volume large and therefore we want to offload some of the water and therefore produce a dilute urine? Now, the side of that homeostasis that this slide is focused on, and the control system that we're first going to consider, is the one that acts to retain or increase the body's water. So in the collecting duct, the goal here is to produce a concentrated urine so that the blood retains water. And we've already talked about how that's achieved with aquaporons, or water pores, along the collecting duct. What we're going to do now is add a hormone that's responsible for increasing the density of aquaporons. That is, making it so that your body's going to retain more of the water. So if we focus in on the epithelial lining of the collecting duct, what we have here is a capillary with this hormone vasopressin, shown in green. And vasopressin is going to act on these epithelial cells. And these are aquaporons. And they, of course, allow water to flow across the cells. And so what we have on the right is the water that's within the collecting duct. And the goal here is to get some of that water into the bloodstream. And we can see, this is some simplification, obviously. We just have one aquaporon on this side of the cell. But in response to vasopressin, the density of aquaporins increases. So the vasopressins move through the circulatory system. There's a receptor for vasopressin. The cell responds to that. It stimulates a second messenger pathway. And that second messenger pathway results in the movement of these vesicles. So the way that aquaporins are regulated is that essentially when they're not in rotation, they pinch off from the cellular membrane and are attached to vesicles. And when they're within the interior of the cell, they don't do anything. It's not until the aquaporins affix to the cell membrane that they are functional and allow water to move into the cell. So that pathway results in more aquaporins binding to the cellular membrane, which allows for the water to traverse the cells, to traverse the membrane, and enter into the bloodstream. Okay, so vasopressin increases the density of aquaporins in the collecting duct, which results in water retention, that is, moving water from the filtrate into the bloodstream. That's going to act to maintain the osmolarity that's in the blood. Okay. Otherwise, if the water were to stay in the collecting duct and produce a more dilute urine, then we would be losing water. The osmolarity of the blood would be increasing. So I've put a little minus here. I mean, it really doesn't reduce the osmolarity, but it helps. This water retention helps to prevent any further increases in blood osmolarity. So that's a little bit more information on the efferent end of this control system that we're piecing together. Let's talk about sensing and integration. So here's where the central nervous system comes in. The hypothalamus, we've seen that the hypothalamus come up before, like in thermoregulation, for example. The hypothalamus plays a role here in the regulation, not just of water, but also salts that are in the bloodstream. And within the hypothalamus, there are osmoreceptors. Receptors, these are neurons that are modified to respond to changes in blood osmolarity. So let's zoom in on the hypothalamus. The hypothalamus is in close proximity to the pituitary. And the pituitary secretes hormones. There's the anterior and posterior pituitary. In this case, osmoreceptors from the hypothalamus span the hypothalamus and span both the hypothalamus and the posterior pituitary. So the posterior pituitary you can think of as being on the business end or the efferent end of our control system, whereas the cell body in the hypothalamus gives us our sensing. Now, the hypothalamus doesn't have the same blood-brain barrier as in lots of regions of the brain. It is instead very sensitive to blood chemistry. And in this case, it is stimulated by changes or the state of the osmolarity in the blood. And let's just consider for a moment how that works. Okay, so what happens here is you can imagine that this cell is surrounded by capillaries. And when the osmolarity of the blood is high, okay, so think a high osmolarity, that means water is going to tend to move into the blood. Okay, so this movement of water is going from the cell body into the bloodstream, because osmosis favors that action, that the water will tend to move towards a higher solute concentration. Now what happens is then the cell body of the osmoreceptor shrinks. It reduces in volume. And that's a response directly to blood osmolarity. That causes a depolarization of the cell membrane. So there are mechanically gated channels that, when the cell membrane reduces in tension, become activated. So that decrease in osmoreceptor cell volume depolarizes the cell. The cell, in response to that excitation, releases this hormone vasopressin. All right, so we see, and we have that little animated there, so the vasopressin then can move into the bloodstream where it is transported. So this is a pretty neat system because within one cell, you have a sensory response and then the efferent action. And so the osmoreceptors serve as both our sensors as well as our integration centers. Okay, decisions are made based on how much volume change there is in these cells about how much vasopressin is released. Now we have more detail on that decision making. The algorithm that controls osmolarity through the osmoreceptor is a little bit different from some of the algorithms that we've considered so far. So first of all, normally, like when we considered thermoregulation, the way that this equation looked was that if the error was less than some negative threshold, then you had some action. In this case, an action is initiated when the error is above a particular threshold. Okay, so that means that even when we're at the set point, then this action is going to be enacted. And the action is the release of vasopressin. And we have quantitative detail on the set point and the threshold in this case. What we have here is the plasma concentration of vasopressin as a function of the osmolarity of the blood. And so here our set point is 300 milliosmolar. Okay, that is the typical concentration of dissolved particles in the blood. So it's on the right of our plot here. But what we can see is that the threshold is 20. Okay, so that's represented by the minimum value on the x-axis. At a value, an osmolarity value of 280, vasopressin starts to be secreted. And as the osmolarity increases, more and more is released. Okay, so vasopressin, according to our control diagram over here, acts to reduce blood osmolarity. Okay, so as we're increasing the osmolarity, it's tending to drive it back, or at least we could say maintain it, right? Because this actually isn't decreasing the osmolarity, but just maintaining the osmolarity. And I guess you could say then that the maintenance function of vasopressin is greatest on this plot at the set point. So that's a little different from other control systems that we've considered. Now, another efferent action that is stimulated by the hypothalamus is thirst. So as your blood osmolarity rises to a high level, you get thirsty. This occurs if you eat salty foods. The salts are absorbed. That increases the density of dissolved particles in the blood. In other words, your osmolarity goes up and you become thirsty. So that's going to cause you to behaviorally seek out water and drink it. So water intake will go up. As a consequence, blood volume goes up, which is going to dilute those dissolved particles. And that is an active means for decreasing blood osmolarity. So if you take more water in to your body, then this osmolarity will go down. So vasopressin really maintains the water content that you have. Thirst drives to increase the water and reduce blood osmolarity. So that's the first control system. So note that this control system is focused on countering high osmolarity. It does not deal with low osmolarity. That is just not the point of this control system. That is instead a feature of our second control system. Now, we're going to again start on the efferent end and then work our way up to the integrating center. And on the efferent end, again, we're going to start with the nephron. And in this case, it's a feature of the nephron that we haven't talked about before. And that is a arteriole that provides blood flow for the glomerulus. Okay, so you remember the Bowman's capsule, and the glomerulus is the site of filtration. So that is the filtrate being produced, and it's being driven by hydrostatic pressure, of course. There, like all the capillaries that we've considered, there's an arteriole that's just upstream of that, so the blood would flow into the glomerulus and then through this vessel in that direction right there. So when the diameter of that arteriole is small, there's a lot of resistance to it, and the hydrostatic pressure is reduced as a consequence of that. Now, there's a hormone, which has a long name, called atrial natriuretic peptides, or ANP. ANP stimulates vasodilation. Of this arteriole. It opens up the diameter of the arteriole, which increases the hydrostatic pressure at the glomerulus, and that generates greater filtration. Okay, so why is that a good thing? Well, if you have ANP creating more filtration through those arterioles, then more water is going to be secreted or excreted in response to that. That is going to have a negative effect on the volume of water that is in the blood, or the blood volume will decrease. And the osmolarity, by the way, will increase as a consequence of that. Okay, so ANP causes a reduction in blood volume. Now, we've already talked about how blood volume influences blood pressure because the major arteries are inflated by blood. We have that injection of pressure from the left ventricle. It inflates the walls of the major arteries like water balloon. And so blood volume and blood pressure are linked in this sense. And the urinary system provides an outflow, and we talked about this before, and we see this illustrated here, provides an outflow of water from the circulatory system. Now, it's not just the major arteries that are inflated by a large blood volume. The chambers of the heart also are influenced. And this is one reason why hypertension is dangerous, is that it strains the muscular walls of the heart. So here we have the heart shown in gray. As the heart beat, it expands, and that expansion stresses the walls, and it requires that the muscles generate more force. So the atria and ventricles of the heart are influenced by these expansions during diastole, and the stress is generated during systole too. Those cycles of stresses in the atria are what triggers the release of ANP. And I don't have all the molecular details on that, and so we'll treat that as a black box. But just be aware that stress in the walls, and that stress can be generated by an inflated blood volume, that stress causes the release of ANP. So it's myocardial cells, and for some reason, the atrial myocardial cells are more important in this function. So atrial myocardial cells sense blood volume through the stress in those walls, and in response to that, it releases ANP. ANP then does all of those things that we detailed in the nephron that seek to reduce blood volume, and it's doing it through filtration. So the question that we set out to address is, how is water volume regulated? We have this control system where an increase in blood volume stimulates water loss through the urinary system, through filtration, in particular in the nephrons. The other control system deals with the other end of the effects of water volume. And that's caused by an increase in blood osmolarity, which would be created by a low blood volume, stimulates an increase in the water volume in the body. So there's water retention through vasopressin and water volume increase by drinking water.